And we're back to the part three, and we were interrupted. So let me continue on, move on to the physiology of digestion and absorption. So let's start with the mechanism of digestion and absorption. Well, we know that digestion breaks down ingested food to their smallest particle that is absorbable, basically in a unit by which we call them building blocks. And only these molecules are small enough really to be absorbed across the wall of the small intestine. So we want them really to be in that particular aspect, very, very small. So digestion is a catabolic process. It breaks down big molecules to its smaller units, small enough for absorption. And you have internal or intrinsic accessory glands and enzymes that are involved in these digestion. The enzymes usually carry out hydrolysis, which means it's a type of digestion that requires the presence of water to break the chemical bonds, or requires the addition of water to help break the chemical bonds. And pretty much absorption is a process of moving substances from the lumen, okay, the inside of the gut into the body itself. See, the gut or the lumen of the gut is topologically still an external environment. Whatever it is that you do not end up breaking or digesting will definitely be going out the other way. Okay, undigested, you're going to be defecating it. You're not going to internalize it. Doesn't get inside your body. Or whatever can be running through or walking through the cells of the walls of the gut, then definitely it ends up inside our body. Tight junctions ensure that molecules must pass through these epithelial cells rather than in between them. So, but you cannot help it if they run in between, most of them will have to go right through. Okay, and pretty much the materials enter the cell through the apical side, which is actually the one facing the lumen. This part here that faces the lumen. Let me turn on my laser pointer. Okay, this section here that faces the lumen of the gut itself is the apical section, and you can see the microvilli. So materials enter the cell through the apical and exits through the basolateral membrane. So this is actually the membrane, the base or the lateral membrane. And then it is already going to go next into the passing right through the interstitial fluid. It ends up in the capillary. Lipid molecules can be absorbed passively through the membrane. However, because of our phospholipid, bilayer phospholipid plasma membrane. However, polar substances are absorbed by active transport. Some nutrients are absorbed before even the chyme can reach all the way to the end of the ileum. So let's take a look at how nutrients are processed. Let's start with the digestion of carbohydrates. Now, with the digestion of carbohydrates, the end product must be a monosaccharide so that it can be absorbed. Therefore, your starch are too big. Even the disaccharides still need to be broken down, okay, to its basic unit, which will be monosaccharides. Digestion begins in the mouth with salivary amylase. So salivary amylase here, okay, sometimes you notice that when you put something like something starchy, like a cookie or a cracker, that's very starchy in your mouth and doesn't have any sugar though, sooner or later you feel that it's becoming mildly sweet. It's because some of the starch is actually being cut into smaller pieces, and the smaller disaccharides and monosaccharides are sweeter than starch. So they pretty much will be broken down into the disaccharide, okay? And the final breakdown into monosaccharides, meaning into its glucose, fructose, and galactose form, is actually going to happen with the use of the um, brush border enzymes, okay? So your lactose gets broken down into galactose and glucose. Maltose will be broken down into two pieces of glucose. Sucrose gets to be broken down into glucose and fructose. And all of this is happening on the final digestion touch that is going to be brought about by your lactase, maltase, and sucrase, or dextranase and glucoamylases. These are um, breaking down disaccharides into monosaccharides. Okay, and they all happen in the small intestine. It starts with in the intestine when pancreatic amylase would break down starch or glycogen that escape the salivary amylase. Anything that's actually like, well, if you have enough salivary amylase, it can break down really a lot of the bigger starch or carbohydrates. But anything that escape the salivary amylase, it's going to be broken down in the what do you call it, in the intestine with the help of pancreatic amylase. Then brush border enzymes will take over for the breaking them into uh, lactose, maltose, and then the monosaccharides in the monosaccharide forms. Monosaccharides are co-transported across the apical membrane of absorptive epithelial cells, mostly by secondary active transport with sodium. And monosaccharides exits across the basolateral membrane by facilitated diffusion. There you go, guys. This is pretty much how the monosaccharides will end up. Okay, they exit across the basolateral. This is the basolateral membrane by facilitated diffusion. Okay, so here it starts with your very big starch. Okay, pancreatic amylase breaks it down, or maybe salivary amylase. Okay, then the brush border, once they reach the small intestine, brush border enzymes along the microvilli will break oligosaccharides and disaccharides into monosaccharides. And finally, those monosaccharides will travel down inside the cell, crossing the basolateral membrane, and then they're going to be transported by way of um, active transport right there. And then they get to be absorbed in the blood, right through the blood. Okay, so some clinical imbalances in carbohydrate digestion. People with lactose intolerance would have very small, will not be producing a lot of lactase and therefore cannot consume lactose because they're not going to be digested. Any lactose that will be eaten remains undigested and creates an osmotic gradient in the intestine, preventing water from being absorbed, and therefore it results in diarrhea. It can also pull water from the intestinal space into the intestinal lumen. Bacterial metabolism of undigested solids produces large number or large amount of gas, sometimes resulting to extreme bloating, flatulence, and really pain. And the treatment for this is actually lactase. So the enzyme is actually going to be either incorporated in the milk, okay, the prepared milk, or as lactase tablets before consuming milk products. So now let's get to how proteins are digested. So proteins are not only getting into our body through its dietary source, but also includes the proteins that are in self-digestive enzymes. They're actually also proteins, and proteins that are being broken down from our mucosal cells. So uh, we actually get protein from our diet, but we also have protein that's part of our digestive enzymes. We have also proteins that are coming from our mucus, and therefore we are digesting them also. They're broken down into large polypeptides, then into smaller polypeptides and small peptides, finally into the amino acid monomers, okay? And pretty much the digestive system will only be absorbing the amino acids. So the absorbable form of protein is actually the amino acid form. So right here it starts with uh, in the stomach where pepsin is going to be produced from pepsinogen in the presence of HCl. It breaks down the proteins into large polypeptides. And then once it reaches in the pancreas, all of those undigested proteins will be acted upon by pancreatic enzymes. And then it will be converting them into trypsin, then chymotrypsin, and carboxypeptidases. And once they are in the amino acid form, okay, they pretty much the amino acid, the brush border enzyme, aminopeptidases, carboxypeptidases, and dipeptidases will help convert the small peptides into amino acid form. They are the ones that are absorbed via co-transport with sodium pump, and some dipeptides and tripeptides are also absorbed via co-transport with hydrogen ion and hydrolyzed into amino acids with the cells. There's also what you call transcytosis of small peptides, and amino acids leave the epithelial cells by facilitated diffusion, and they enter the capillary blood in the villi and are transported into the liver via the hepatic portal vein. Okay, here it is, guys. That's your protein. Okay, pancreatic proteases will break this big protein into polypeptides. Once they are broken, the brush border enzymes will break protein fragments into smaller amino acids. Then your pretty much the amino acids are co-transported across, okay, the apical membrane of the what do you call this, the epithelial cells. And then this active transport uses sodium ion concentration gradient that is established by the sodium potassium ATPase pump right here in the basolateral membrane. That's actually your ATPase pump. And finally, your amino acids will cross the basolateral membrane and will now be entering the capillary. It's going to be brought into the liver via the hepatic portal vein. So here is actually the third, and hopefully not the last because we have nucleotides. Now, the third in the digestion process, the big digestion process, is the digestion of lipids, which is kind of like unusual because it has a different route by which we digest them. So the first step in lipid digestion in the intestine is to emulsify it. Remember that lipids are fat. And being that our lipase is a protease, that rather a lipase rather, our lipase is actually a hydrolytic enzyme, it would require water. And you know that lipids are not soluble in water, so it needs to be emulsified. Triglycerides and the breakdown products are insoluble in water, so there needs to be a treatment with bile salts that break large fat globules into smaller ones. Then the next step will be digestion, where your pancreatic lipase rather breaks down fat into monoglyceride and 2 free fatty acid. And then the next step will be to form micelle. Now, this means that the product from digestion becomes coated with bile salts and lecithin. Then finally, the lipid products would diffuse would leave the micelles and cross the epithelial membrane via diffusion. So pretty much, uh, the chylomicron is formed after that, and lipid products are converted back into triglycerides and packaged with lecithin and lipoprotein. And they are this aid at this end, they are actually forming what you call a unit called chylomicron. Chylomicrons are now going to leave the cell by exocytosis from the basolateral side, and it enters the lymphatic lacteals. Eventually, it is emptied into the venous blood at the thoracic duct, because the lacteal is is actually a lymphatic duct. So together now with the lymph, it is now going to be emptied back into the venous blood via the thoracic duct, emptying into the left subclavian vein. Okay? So, here you go, guys. This is your fat globule. Start with the fat globule. And pretty much your fat globule, after it's acted upon uh, by bile, gets to be emulsified in the duodenum, breaking them into small fat globules, okay, in smaller fat droplets, increasing the surface area for the lipase enzyme to work on them. Now digestion is going to happen. Pancreatic lipase will hydrolyze the triglycerides, yielding monoglycerides and free fatty acids. And now micelles are actually formed. These are actually made up of fatty acids and monoglycerides and bile salts. They ferry their contents immediately into the epithelial cells. Then this is already your villi, and you have your epithelial cells right there. The micelles now will be diffusing. Fatty acids and monoglycerides diffuse from the micelles into the epithelial cells, and you could see them right here. Those are already the fatty acids and monoglycerides. These fatty acids and monoglycerides now will be recombined again and packaged with other fatty substances and proteins to form what you call chylomicrons. And these chylomicrons, those triplet units right there, are extruded from the epithelial cells by exocytosis. They enter the lacteal. This tube here is the lacteal. It is actually a lymphatic duct. And they are carried away from the intestines via the lymph into the thoracic duct, and then into the left subclavian vein. So now the last of the macromolecule. Let's not forget that there are 4 macromolecules that makes up a living organism, most especially us. You have the nucleic acid, our DNA and RNA, and the food that we are eating, being that they're coming from living organisms, are actually or have a lot of nucleic acids in them. So they do contain DNA and RNA. We have pancreatic nucleases as part of the pancreatic juice that would hydrolyze nucleic acid into nucleotide monomers. Then, as soon as they're broken down, brush border enzymes will take over. These brush border enzymes are the nucleosidases and phosphatases. They break nucleotides into free nitrogenous bases, into pentose sugar, and into phosphate ions. So your nucleotide needs to be broken down further into their smaller subunits, which will be your nitrogenous bases, your adenine, thymine, cytosine, guanine, and uracil, and pentose sugars, or the pentose sugar, the oxyribose and ribose sugars, and the phosphate ions. The breakdown product eventually will be transported by special carriers in the epithelium of the villi. So we're winding down and getting into the absorption of vitamins and electrolytes and water. So vitamin absorption happens in the small intestine. Your fat-soluble vitamins, such as your A, D, E, and K, are carried by micelles. just like any other fat, they diffuse into absorptive cells. The water soluble ones, which will be your C and B, are absorbed by diffusion or by passive or transport or active transporters. B12, which is really pretty large and is actually charged, binds with the intrinsic factor. That's why we need the intrinsic factor that is produced by the parietal cells because of the extraordinary large size of the B12, and therefore is absorbed through endocytosis. So in the large intestine, vitamin K and vitamin B from bacterial metabolism are also absorbed. So absorption of electrolytes, your sodium, your potassium, okay, these are ions, are transported actively along the entire length of the small intestine. Your iron and calcium are so absorbed in the duodenum. The sodium is coupled rather with active absorption of glucose and amino acids. So there is a co-transport there, an antiport. Then chloride is transported actively. Your potassium diffuses in response to osmotic gradients and is lost if water absorption is very poor. And the usually amount in intestine is pretty much the amount that is absorbed. So iron and calcium absorption is related to need. Therefore, ionic iron is stored in the mucosal cells with ferritin, and when needed is transported in the blood through transferrin. Calcium absorption is regulated by vitamin D and the parathyroid hormone. Water. About 9 liters of water, mostly from the GI tract secretions, would enter the small intestine. 95% of it is absorbed in the small intestine by osmosis, and most of the rest is absorbed in the large intestine. The net osmosis would occur if concentration gradient is established by the active transport of solutes. Water uptake is coupled with solute uptake. Now, poor absorption or malabsorption can be caused by anything that interferes with the delivery of bile or pancreatic juice. And this could be due to damaged intestinal mucosa, maybe from bacterial infection, or maybe some harsh effects of antibiotics. For instance, for people who are gluten sensitive, they say that they have allergy to gluten. It's actually a celiac disease. This is actually common malabsorption disease. It's an immune reaction really to gluten. Gluten causes the immune cell damage to the intestinal villi and brush border. And usually the treatment for this is to eliminate gluten from the diet. We are now finished, and I bid you all well and happy studying. You're going to have your next test in November 5, which is Thursday of next week. November 5, 2020.